platform brings together experts to share the insight, research, innovation, and foster a collaborative ecosystem to transform patient care and improve healthcare outcomes. Today, we have the journey of discovery and exploration as we collectively strive to revolutionize healthcare and build a brighter future for patients and communities.
Sri Pharmacy was established in May 1997 by well-known philanthropist and industrialist late Sri Padma Dr. P.V. Raju and the P.V. Raju Foundation and Swedish Educational Society. Sri K.V. Vishnu Raju Garu and his grandson succeeded as the chairman and is making all efforts to impart quality education. Sri Vishnu Raju Pharmacy is the first private college affiliated to Andhra University Visakhapatnam. Our campus is spread over 108 acres with latest equipment, spacious air-conditioned smart lecture halls, computer labs, and seminar halls, and with good library facilities. The Vishnu Pharmacy is only recognized pharmaceutical research center under Andhra University. Our faculty members got research grants from CSIR, DPT, DST, etc. SPCT is the first pharmacy college in Andhra Pradesh affiliated Bangalore, and our B2 program is MBA equivalent.
To begin the program, now I call upon our honorable principal, Dr. K.S. Nagaraj to take the center stage.
Before that, now let's have a small glimpse of our college.
What is that?
I request one of my friends to enter the voting.
Explain that.
Now I request Dr. A.D. Rasikiran sir, Professor of Department of Biotechnology, Andhra University, to come on the dais. I request one of my friends to address the audience. B. Raju Institute of Technology, Nagpur, Hyderabad, to come on the dais. I request one of my friends to address the audience. Welcome.
I request everyone to join us in this gathering. I request Manoj Young for the prayer song.
And it was everyone to please stand.
నెంబర్ ప్లేట్ మార్చుకుంటారా నేమ్ ప్లేట్ అది చెప్తాను మహాలక్ష్మి ఒక నేమ్ ప్లేట్ చెప్తాను మార్చి
The most important is the students is overall development. And in that context, participation of seminar or workshop, when it's happening outside your college, you always have an advantage to make you develop yourself. Okay? So this is officially today's team is revolutionizing healthcare, integrating pharmacy, bioinformatics, and analytic technology. This is one of the emerging fields the team present world of science. Because everybody wants that to, because healthcare is delivered in the government and so one of the first area as far as concerned for healthcare professionals is bioinformatics. So this was observed by our vice chairman very long time, and he told that we had to motivate the students to focus on this bioinformatics. Okay, we are pharmacy graduates. I think some of the students who are going abroad, they're mostly preparing bioinformatics. So that we take this team to take forward and much more discussions to be happening in the future. In that, I'm very thankful to the Dr. Rajan and Dr. William Sharma guys. Okay? So because they both of them they come forward, they accepted our invitation, and invitation for that I'm very thankful to madam. So when attending for this kind of seminars or workshops, some takeaway points are most important. I hope you get many more takeaway points from this session, and they will have more moments to be added. Thank you very much once again, and congratulating all the participants and delegates of this seminar, national seminar of program 025. This is the brain state of Promilan is the former director Dr. Vasuraj Garu. He is the founder principal of this institute. He made this program, but the legacy of the program is here continuing. Okay? And very thankful for the management, chairman sir, and vice chairman sir, and admin staff, provided necessary facilities for the students for better development of the students. So you might have seen many campuses across the nation, But uniqueness of this campus is that the integration among all the colleges is very essential here. Because the person who is studying at pharmacy college, they can use the facilities of the dental college. And dental college students can make use of the pharmacy. Similarly, we are collaborating with the college. That is the one of the unique feature of this the society, Christian education society. Okay, so it is in a bigger way, these are our responsibility on what we are right. So definitely with all your support and blessings of Padma Dr. Devi or the founder chairman of this Christian education society. Once again, warm welcome to on behalf of the Christian College of Fountain, the Christian Education Society and Dr. Devi Foundation. Thank you all. Thank you.
Thank you, sir. Now I request the dignitaries to take their seats on the stage.
We have scientific presentation by Ladies Radican Sir, Professor Department of Biotechnology and the University. Third call on Manju Bharti, clear each introduction about today.
వీడియో కూడా మీకు పెట్టగలరు వాట్సాప్ మెయిల్ పెట్టేస్తారు ఏదైనా
Good morning, God. It was distinct honor to Dr. Ravikiran as Editor, education scientist, educator, and entrepreneur with a truly remarkable career. Dr. Ravikiran's academic journey is marked by excellence, holding a PhD in biochemistry and molecular biology with a minor in pharmacology from the Washington State Medical School, Detroit, US, where he also earned his MS in molecular biotechnology with a minor in genetic engineering from Washington State University, US. His foundation education includes an MSc in biotechnology and a PhD in biology and chemistry, both from the Andhra University in India. Dr. Ravikiran's research project is evident in his extensive work on over 50 biomedical research projects funded by prestigious institutions like the US National Institute of Health, the Canadian Institute of Health Research, and NPT India, and startup firms. His contributions to the scientific community are further highlighted by more than 82 research articles, where 26 articles are physically published and 51 other articles, and a portfolio of 17 patents showcasing his innovative skills. His achievements are being recognized with multiple awards and honors in the USA, Canada, and India, including the Quality Innovator Award for having the highest number of patents in the year 2023 to 2024. International Cancer Research Training Award from the US Federal Government, Washington, DC. Best Oral Presentation Award in Medicinal Chemistry and Drug Discovery, Detroit, US. International Research and Training Assistant Award from Washington State University, Detroit, USA. He has held significant positions, including Professor of Practice at Andhra University, Science at the DBT India, Structural Biology, and a Cancer Research Scientist Fellow at the US Navy Cancer Institute. He is also a recipient of multiple honors in USA, Canada, and India. Those are felicitation honors received from the Vice Chancellor of JNTU, Kakinada, visiting professor in the Department of Zoology, Andhra University. He is also felicitated as a guest speaker for the National Council League, College of Pharmaceutical Sciences, Andhra University. Guest of honor presentation for delivering the popular lecture sponsored by the Department of Biotechnology, Government of India, organized by Garden City University, Bangalore. Dr. Ravikiran is also a dedicated educator, having mentored nearly 2,000 students in research laboratory science. He has served as a professor of practice, guest faculty, and visiting professor at Andhra University, imparting his knowledge and passion to the next generation of scientists. His entrepreneurial spirit is demonstrated by founding of two startups, which are the IIT Institute of Discovery and Education, Toronto, in Canada, and the Center for Advanced Applied Biological Sciences and Entrepreneurship in India. Dr. Ravikiran's expertise is widely sought after, as evidenced by his more than 50 national and international invited talks given in India, USA, and Canada. He has also initiated 20 national and international research collaborations. He also deposited 36 excellent digital structures in the 13 data bank, and also organized multiple conferences, symposia, and colloquia in US and English. So please join me in welcoming Dr. W. S. Garden.
Thank you all for giving me this wonderful opportunity to share my thoughts. First of all, I'm very highest regards to the author of this video. Government has given us a quick tour of your college today, and I'm extremely impressed by the way of your college is built and organized in different blocks. And today, I'm going to keep it very short. I'm going to discuss a few things on the latest findings that we did in our lab using bioinformatics and computational biology. My lab is a startup company. It is actually creating the Andhra University campus. And so most of the projects that we do in our lab, they're multidisciplinary. So we do anywhere from how to design a drug or how to test a drug, the regulatory affairs, and there will be post-marketing surveillance, vaccine design, genetic engineering, and the latest breakthrough that we did in our lab is my students they were able to isolate endometrial stem cells from the menstrual waste, and they were able to 3D print into small shapes. So this got very high impact media attention. So before I get started with my scientific presentation, I'd like to share a few accomplishments of my students in my lab. And I guess I made a small video. I'll just wait for the video.
One second, just 2 minutes. Okay. Yes. Alright.
Thank you. So that's a very quick, brief overview of our latest findings, and I'm very proud of each of my students. They filed for patents, and we're actually thinking of starting companies based on the discoveries or inventions that we did in our lab. And now I'll get started with my presentation.
Thanks. Okay.
Okay, so I'm going to go through briefly on the project where we worked on the human CYP3A4. Most of you are familiar with the cytochrome P450, the 3A4 class. That's the most important enzyme present in your liver primarily, and it's involved in metabolizing a lot of drugs, a lot of different drugs actually. So we wanted to know if any of the latest COVID-19 medications that were approved by the FDA under the emergency usage. So there's a criteria
Slide and
called the EUA, emergency usage authorization. So the FDA approves certain drugs and vaccines, in the emergency situations like the COVID-19 pandemic, where you're trying to save lives. So I'm going to talk to you about the story of one of those such drugs, molnupiravir. So the molnupiravir was approved by the FDA for usage under emergency only. It's not approved for regular usage. So as you all know, COVID-19 has been a very big pandemic, and millions of lives were lost across the world. We competed with each other, right? So many countries, India competed with the United States, so did other European countries. But it's very unfortunate that, we have all this latest technology, but still we lost millions of lives during a short period of time. That's probably within 1 or 2 years. But this kind of situation is actually challenges scientific discoveries, where everybody now is focusing on the mRNA vaccines. So mRNA vaccines were not popular before the COVID-19 pandemic, but now they've become very popular. So it's not only the vaccines, but there are also antiviral drugs. I think I'm showing a picture in the next slide. Yeah, so there are 3 drugs that were FDA approved: remdesivir, molnupiravir, and tenofovir. So remdesivir is the only one that was properly approved with the appropriate efficacy and the full pharmacological pharmacotox data. Molnupiravir and the tenofovir, they had only a few data points, but the data was good enough for the emergency approval for the treatment during the COVID-19 pandemic. And also on the right side, we can see the green color shows a chimeric spike protein of the coronavirus. And the business end of the spike protein is the receptor binding domain that's highlighted in the blue color on the side and zoomed into a small box. So that receptor binding domain is a critical part of the spike protein because it helps binding the human ACE2 receptor during the infection process. So this RBD was used for the vaccine design in most of the cases. So today I'm not going to talk about the RBD studies that we did. I'm going to focus mainly on the molnupiravir that was approved by the FDA under EUA. So molnupiravir is actually it can be orally administered. So it is a prodrug that resembles a nucleotide, and it can morph itself into either a cytosine or uracil. You can see the same drug can pose itself as either cytosine or uracil. So because of this, it can create mutations randomly. So the proposed mechanism of molnupiravir is that the virus, when it undergoes replication rapidly, molnupiravir will rapidly induce mutations by posing itself as a C and then later changing into a U, or posing itself as U and later changing into a C. So when the drug generates so many mutations in the viral genome, the virus becomes very less effective because the virus is full of mutations. But sometimes, unfortunately, there were viral strains that were selected because of this situation. It's called clinical evolution. Okay? And then, as you can see, when you see the DNA double-strand paired DNA replicating a couple of rounds, you see all these mutations getting accumulated. This work is already published, so you can actually look it up online. So, what if, we give this molnupiravir to patients, that have COVID-19? So that's the first question that we wanted to see. And then, in addition to this, we are also interested in knowing how does this, work in the case of a patient who has COVID-19 plus other complications such as diabetes, hypertension, which are, quite common these days because of the stressful lifestyles. And the third one is, how does this play a role in a patient with all these complications and under immunocompromised situation? So, we want to focus primarily on the CYP3A4 because CYP3A4 is the primary drug metabolizing enzyme, not only for molnupiravir, but also for other drugs. And on the first slide, I showed you three drugs. One of the three drugs is dolutegravir. So the dolutegravir is actually a combination with ritonavir. So ritonavir is a CYP3A4 inhibitor. And I'm going to talk about that today. But I'm just saying, CYP3A4 plays a very important role. So that's why we wanted to see how these mutations or the mutations induced by molnupiravir, how do they affect the function of CYP3A4? Today, I'm just going to focus only on the bioinformatics and computational biology aspect of this project, because I think the focus is on the bioinformatics. But we're actually going through the wet lab experiments of the week. Okay, so when you talk about these cytochrome P450 enzymes, no matter what kind of CYP it is, it always contains a heme 4-5 ring. As you can see, that's the heme 4-5 ring with an iron atom in the center. And in the human CYP3A4, you can see the heme ring is interacting with 5 critical amino acids. 4 of them are arginines and 1 is tryptophan. So these 5 amino acids are very critical to actually bind to the heme 4-5 ring and keep it in the shape. Because any changes in the conformation of these heme 4-5 ring will affect the function of the CYP3A4. Whether it is oxidation, eruption, or any of the simple reactions that the CYP3A4 is involved in. Okay, so from now on, I'm going to talk about MIMS, molnupiravir-induced mutations. So what we did was we performed a bioinformatics study. So we've taken the entire gene sequence, the mRNA sequence of the CYP3A4, and then we calculated all the possible mutations that the molnupiravir can induce in this particular mRNA of the human CYP3A4. And we came up close to about 1,000 different types of mutants. But that's only talking about single mutation. We haven't done the combinations yet. We have done a few combinations where we've identified two mutations, 2 names at a time or 3 names at a time. But today I'm just going to talk about how these single names affect the function of the CYP3A4. So what we basically did was we took the structure of the wild type CYP3A4. So the wild type CYP3A4 meaning it doesn't have any mutations. And we modeled these MIMs into this protein, and then we performed molecular dynamic simulations to relax the system to see how these MIMs will affect the binding or the stability of the E4-5 ring. Because, like I mentioned earlier, the spatial orientation of this E4-5 ring, the active site of CYP3A4, is very critical when it comes to the metabolism of different types of drugs. So, in my lab, I encourage art in addition to the science. So every time my students, when they do these molecular dynamic simulations, there are different types of outputs. So typically, molecular dynamic simulation gives you the C-alpha, RMSC, and the root mean square fluctuations of amino acid side chains and the backbone of the protein, and also the number of hydrogen bonds made by the CYP with the heme ring, then the van der Waals contacts, so we have all this data. So every time we have a big data set, I mean, it's not a large data set, it's a big data set for our analysis. I always ask my students to draw these circular plots, and I want to see the plotting of all the data in a single diagram. Believe it or not, here you're actually seeing a hand-drawn circular plot. So this was actually drawn by hand, and this is not printed. Okay, so it's not prepared using any graphics. So this hand-drawn circular plot explains to you how each MIM will affect the CL or MSC or the shape or the structure, manifold deviations in the city or during the molecular dynamics simulations. Okay, so we found that, some of these mutants, if you do the NIMS, they indeed lost contacts with the E4 brain. So typically, like I showed you earlier, if you want to maintain the E4 brain stable, you have to have all those 5 amino acids maintaining their contacts in the active site of the CYP4. But when you have one of these NIMS, for example, you can see here, W126Q, that's the first red bar on the left side. And then R375G, that's the middle red bar. And then the third one is S437D. So these mutations, they actually led to loss of contacts with the E4 domain, which means these things will definitely affect the function of CYP4. So in addition to this, there is one more thing that I want to mention to you is that molnupiravir is able to induce premature stop codons. So because it is inducing the mutation, it is also capable of inducing premature stop codons. So for example, let's say the length of your gene is this much, and if there's a stop codon in the middle, that means your final protein will be smaller than the actual expected size. Right? So that is called a truncated protein. Next slide, please. So here, I'm showing you the comparison between the wild type and the mutant. As you can see, there's a lot of contacts. So the number of hydrogen bonds, previously the wild type used to be 6 to 8. And in the mutant, we only find 4. So there is a lot of contacts, the hydrogen bonds, they team more iron. Next slide, please. And when we perform the molecular dynamic simulations, as you can see, the E4 ring in the mutants, it actually has tilted. Okay, so on the right side, we can see in the magenta color, that's the mutant, where the E4 ring is not horizontal anymore, it's actually tilted 14 degrees. So because of this 14 degrees tilt in the E4 ring, you can see the inhibitor, the ritonavir that was bound to the E4 ring, also had shifted its orientation. So on the left side is before the molecular dynamic simulation, where you can see both the heme rings are perfectly overlapping each other, and both of them are horizontal. But after the molecular dynamic simulation, you can see the mutant one is tilted. So that tells us that happens because of loss of contacts. Next slide, please. So in this figure, you can clearly see before the molecular dynamic simulations and after the molecular dynamic simulations. So the mutant causes, or sorry, the loss of contacts causes tilt in the E4 ring, and because of that, the entire ritonavir molecule, which is the inhibitor, is tilted much bigger. The angle of the tilt for the ritonavir is much higher compared to the angle of tilt for the E4 ring. So this diagram clearly depicts that. Okay, so as I mentioned earlier, the premature stop codons. So as you can see on the left side, you have the wild type CYP4. It's a big protein, right? But when you have a premature stop codon, what happens is the length of the protein is truncated because of the premature stop codon. So the length of the protein is truncated, you can see the size of the protein has significantly decreased, right? So the wild type is a full protein, which is very big protein, but the mutant that has premature stop codon is a very small protein or truncated protein. In other words, the truncated protein does not even have the E4 ring. So imagine if you have a CYP3A4 without the E4 ring, then it is totally useless. Next slide. So based on this preliminary data, we conclude that the NIMS, molecular induced mutations, they cause significant damage to the overall treating structure of the CYP3A4. And the CYP3A4 is one of the critical enzymes for the drug metabolism in humans. The premature stop codons induced by this NIMS actually cause significant loss of the function of the CYP3A4 because the protein is significantly truncated. In addition to these, we also, I also wanted to emphasize, like, the drugs that are approved by the FDA under the EUA, they're not always perfect, okay? They're supposed to be used only for the emergency situation. So if you use molecular on a daily basis, then that will potentially cause a lot of problems. A few of them I have actually showed here. So the take home message I would say is, take the drugs only under the physician's prescription, physician's advice, follow the physicians. And don't make yourself decisions. And whenever you have any medical problems, please don't go to the medical shop. You have to go and see a doctor. Because the doctor will prescribe, and then you go to the medical shop. But most of the times, I think the regular habit that I see with people in India is they go to the pharmacy first, they go to the medical shops first, and say, I'm not calling about marketing. That's all we do, right? Then obviously, they give you some tablet, we take the tablet, and that's the end of your story. So please don't go to the medical shop if you have a problem, medical problem, go see a doctor first and then follow the prescription. Otherwise, you have consequences to face. So I just wanted to, give you one of the productive stories from my lab. This is my student, Madhuri. What she did was she genetically modified lactobacillus bacteria. So that the bacteria will produce small pieces of spike protein from coronavirus. So she took these genetically engineered lactobacilli and prepared yogurt using milk. And you can just take this yogurt, you can eat the yogurt. So what happens is when these lactobacilli go to your intestines, they produce spike protein and the immune system gets alerted. So it's like vaccination. And you can eat whatever amount of yogurt you eat. And if you want to eat 2 cups, you can eat 2 cups. If you want to eat strawberry flavor, chocolate flavor, it's up to you. So you don't have to get the shots and booster shots and get swellings. You can just simply eat yogurt and you get vaccinated against COVID-19. So we filed for 3 patents on this invention. So we are actually getting ready to start a company based on this YoVac. So we named this as YoVac. So it is yogurt vaccine. And there's another YoVac that we're working on. It is made with almond milk. Because some people are lactose intolerant. So for the regular yogurt, some people are lactose intolerant. So we're using almond milk. So we're preparing almond yogurt vaccine. So that can be consumed by people who are lactose intolerant. So just wanted to give you a quick, summary of what we have done in terms of protecting people from the COVID-19. And with that, I thank everybody. I thank to President sir, and thank you all for giving me this opportunity and the founders to share my research with you, and I'll be happy to take any questions. Thank you. Yeah. So we are working on animals now. And once we get positive feedback, then we will be officially connecting with clinical trials. And then we also want to bring in the flavors. At the moment, we only have regular yoga, but when it comes to the human clinical trials, we also want to try out vanilla, strawberry, chocolate flavors.
Any questions from audience, please? The yoga for the for corona, corona. How far it enters into the systemic way because the virus is directly attacking the respiratory system, not through the gastrointestinal tract. However, this yoga will be absorbed towards the development of the immunity in the body.
So like I said, we are still doing the animal studies. We have complete in vitro results. We have proved that it works in vitro. We have published the paper, so it's available on PubMed. So as soon as we are focusing on the antibody titers right now. So we're trying to see how much immune boosting can it do. So and then another thing that I actually did not mention is this vaccine, the Vac, is self-sustained boosting system. Because the lactobacilli carrying the spike proteins, it goes into the gut microbiome, and then it will transfer those genetic information to other bacteria in your microbiome. So every single bacteria in your microbiome starts producing the coronavirus spike protein, so that acts as a self-boosting system to leverage gene transfer. So I guess the answer to your question is we are still in the animal testing and we want to confirm what kind of antibody titers we have. So far, the results are promising.
Any questions from
Peter Knight, sir, my question is, have you ever checked any interaction with the probiotic with the seed protein?
So therefore, so this probiotic is producing the spike protein. So the spike protein will act as an antigen, right? So spike protein being a protein, it has nothing to do with CYP3A4 because CYP3A4 primarily metabolizes the xenobiotics such as drugs.
But the probiotics have a protective effect when compared to the other bacteria. The drug.
Probiotics have protective effect, and that's a very complicated question because they have very different varieties of protective effects. And how do you
Question is that you have read your insight about the interaction of probiotic with the cytoplasm.
see, the probiotic is a bacterial cell. So I guess I'm not able to understand your question. The probiotic is a bacterial cell that carries genetic information to produce the coronavirus protein. And the CYP3A4 is an enzyme that metabolizes drugs.
If we use the probiotic as a carrier, can the probiotic is a one of the drugs?
I don't think so. No. Because, again, it depends on the context. If you are talking about an immunocompromised patient that has COVID-19 and other complications such as diabetes, hypertension, then no matter how much yogurt you're eating, if you take molnupiravir, then you're destroying your CYP3A4. So the probiotic is helping you to save from the viral infection. But the molnupiravir is actually destroying your CYP3A4. So you're doing a self-destruction by taking molnupiravir. So I would say the suggestion would be when you're taking the Yovac, you should avoid using any antiviral agents such as the molnupiravir and other drugs.
So one more insight I want that how the probiotics will interact with the microbiome.
That's a very good question. So when you're choosing the probiotics, you should protect the probiotics from your immune system because the immune system kills these bacteria, and there's no use, right? So that's why we follow the lactobacillus. And humans have been, especially the Indian citizens, have been eating yogurt for millions of years, right? So everybody eats yogurt, like most of us eat yogurt or drink buttermilk. And we never had any issues with the immune system, so we made a simple assumption that the lactobacillus bacteria were very friendly with the human immune system, so we should be fine.
I mean, this is not infected, that's
No, they are actually not targeted by the immune system.
Any questions from audience, please? No questions? Thank you.
Thank you.
For your valuable presentation and rapid insights on moving packages induced mutations in human CYP protein. Now I call upon Dr. K. Snapchat to step on the dais and on Dr. A. Reference to spoke on the presentation. I request Dr. A. Reference to come on the dais as well. We have another scientific presentation by Dr. Neelam Sharma, Assistant Professor from BVRO Institute of Technology, Nagpur, Hyderabad. I call upon Akia to give a brief intro about knowledge.
Same folder. I want to see whether it's working.
It's my pleasure to introduce Dr. Neelam Sharma Garu, who is currently working as an assistant professor in the Department of CSE at ADR Narsapur. She holds a bachelor's and master's degree in biotechnology and a PhD in computational biology from Philadelphia, Delhi. She pursued her postdoctoral research at the University of California, San Francisco. With over 20 publications in high impact international journals, her research has garnered more than 1,200 citations, achieving an fetch index of 40. She has developed multiple databases and machine learning based predictive tools. Her expertise lies at the intersection of machine learning, AI, and computational biology with a focus on genomics, cheminformatics, and immunoinformatics, particularly in protein or peptide therapies. With great respect and admiration, we all welcome Dr. Neelam Sharma Garu. We are delighted to have you here and excited to learn from your experience and wisdom. Thank you for having me on this presentation for inviting for the talk. I really appreciate to share the stage with prominent scientists working in this area. I am very much appreciate the Dr. Ravi's work and how this these video coverage is actually a part of Indian like, Indian diet, how we can use and eat it as a vaccine rather than injecting it into our arms and then getting fever, then getting these. So, yeah, so what do you want to do is, talk today. My topic is all about the digital pharmacist. Why bioinformatics is your next big skill? Why do you think that bioinformatics is actually a necessary part of today's world? So, in today's scenario, we all are working in the disciplinary area, pharmacy, AI, ML, and health informatics. So, and trying to convey that why bioinformatics is very much necessary for everyone sitting in this room. So, can you show me? Just to give you a brief introduction about my own journey, what background that's taken actually. So, all about my journey. So I completed my bachelor's and master's in biotechnology. If you know that what biotechnology is all about, Professor Ravi has already given a brief introduction about biotechnology. After completing my master's, I was awarded just a prestigious fellowship called DST Inspire. DST Inspire is provided by the Ministry of Science and Technology, and it is given to the people who are first rank holders of the university gold medalists, and only 1,000 students in India get that in a year. So I was privileged to get that. So after taking this fellowship, I enrolled myself in a PhD program. So being from a very lab experiment is how I transitioned into PhD computational biology. During my biotechnology program, there was one semester, in one semester there was a course called bioinformatics, which has actually intrigued me a lot. Wanted to see that how the data generated by experimental values are actually being analyzed. So this intrigued me and then completed my PhD. PhD from IIT Delhi, actually, in computation biology. After completing my PhD, then I moved to University of California, San Francisco, where I conducted a doctoral research. There I was working in genetics and transplantation, there was laboratory, and after coming back from US, I have joined at least. I have joined at least. That's a currently I'm the leader that's support in Department of Science and Technology AI. So starting from biotechnology to computer biology, right now computer science, which in many cases upscaling my constantly upscaling my skills and here I'm in front of you to present this software. Okay, please. Okay. Digital councils, why I'm trying to transforming council data driven thoughts. So as we all know, the pharmacists are not only the people who develop the drugs, but nowadays it is very important for each one of us to know, how to analyze the data which is developed by the government, or develop great data. Yeah. You have to understand the digital transformation in pharmacy, and what digital transformation in pharmacy is all about. So you can see in this figure that in ancient times, when we have Ayurveda for treating our medicines, for treating our diseases, the Ayurvedic, that time they used to call these doctors, they used to give us these herbal medicines, more of a festival and we gave it, they apply onto the skin, and then this is how we get our disease treated. So from the ancient time moving to today's time in 21st century, that the importance of custom-made medicine becomes the most relevant. This is where the bioinformatics plays a very important role. So imagine a world where the drugs are actually personalized. It is made specifically for you, based on your DNA, based on your protein expression. So that is why bioinformatics plays a very important role. Next, please. Then, why bioinformatics? What is bioinformatics? Bioinformatics is an application of computation tools to manage, analyze, and interpret the data. These are the various key components I have carefully picked up for, but there are many. Genomics, proteomics, drug discovery, clinical data analysis, personalized medicine, which is actually a very peak research area these days. Next, please. Role of bioinformatics in pharmacy. Drug discovery, which you all deal about. Drug discovery, computation screening of drug candidates, pharmacogenomics, clinical trials, and less. AI in pharmacy is automating drug interactions and optimizing prescriptions. So we all have to understand that in current pharmaceutical situation, learning and gaining the expertise of AI and ML in every field is required. So don't hesitate or don't have fear that how we people coming from pharmacy background, we will learn AI and ML. If I can do it, you all can do it. Next, please. This is what shows that how mathematics is related to pharmacology. The target identification and validation, prediction of toxicity, personalized medicine, drug repurposing, big data analysis, all these are a big part of pharmacy, which is actually done only using bioinformatics skills. So prediction of toxicity is one of the projects which I also did during my PhD program, and we'll see about it later. Next, please. Now, moving on to the main topic, machine learning in pharmacy. So I wanted to know from you people, how many of you have experience in machine learning or artificial intelligence? If you can raise your hand. How many of you really wanted to learn how to use machine learning and artificial intelligence in farming. I would like everyone of you to raise their hands because in the current market situation, what it has been predicted that 10 years down the line, healthcare informatics is multi-million-dollar market. So we have to upskill, we have to learn these skills if we really want to get into the market and want to get good jobs with good practices. Next please. So a brief introduction about what machine learning is. Can you move forward please? Next. Machine learning, as the name suggests, as we learn, machine learns. How we learn, our teachers make us learn, our teachers give us the syllabus, they give us the lecture, we learn. This is how we make the machine learn. We give the machine the data, machine trains on that data and then learn, and then we validate it, that whether the machine has learned it in correct form or not. So it is the application that provides the system the ability to automatically learn and improve. This from experience without being excessively programmed. Yes. And it focuses on the development of computer programs. I know we people being from biological sciences are very much afraid of using computers, but trust me, computer programming, these all are our friends. We should at least have friendship with programming, it will going to help us in the long term. Next, please. The primary aim of machine learning is to allow the computer to learn automatically and adjust actions accordingly. Next please. Daily examples of machine learning. We all are surrounded by machine learning, artificial intelligence examples throughout our day-to-day life. Like smart searching, Google Map, it uses machine learning and AI for that. This is the paper. It has a lot of animation. Smart speaker, yeah. Alexa. How many can Alexa or Google? We all know. How are they doing? Machine learning. Next. Social media advertisements. How many of you know that Facebook, YouTube, there is any search for one specific term there, and you start getting those leads. Machine learning. Did you learn to train that model? No, it trains automatically. That's what machine learning is all about. Next one. Next. Self-driving cars. I hope we all are aware of self-driving car concept. We don't need drivers these days, we need self-driving cars. So all these days are examples of we using machine learning. Next. Now we will say that how machine learning will be used in the case of pharmacy. How many people know protein? I hope that you all should raise your hands. I believe that you all are pharmacy students, you should know protein. Protein prediction, functional analysis, geoinformatics, and drug discovery, personalized medicine. All these are actually handled by machine learning. We will be seeing one of the examples I have quoted here. Can you go next, please? Yeah. So we will see that how machine learning model is made. This is just I'm touching the overview to give you the overview of that how we build the machine learning model and whatever the data that we work on. So let's say proteins. So there are 5 simple steps, starting from data collection, feature engineering, data modeling, performance evaluation, and then deployment. These are the 5 crucial steps required for developing any machine learning model. And this is how at the back end we can say that as a pharmacist, we are developing our bioinformatics skills. So data collection from these resources, then we compute features, we start building models, then we analyze the model and we deploy it in the form of I'll tell you the data for the facts. Thank you. Start with data collection. So I hope you all are very much aware of the central dogma. Central dogma. So let me correct me if I'm wrong. So DNA translate to protein and then translate to RNA, right? Right. Yeah. DNA translates to protein and then transfers to RNA, right? RNA. So I hope you all are aware. Awake. Yeah, anyone is saying you had breakfast? Okay, I can't say too much, but I have done dinner. So you are that. There. So DNA transfers to RNA then translate to protein. So we all know that DNA nucleotides A, T, G, T. Say, so as RNA, A, U, G, T, and protein is made of time to go beyond of the body. Yeah. So yeah, DNA, RNA, protein. Next please. Data collection. How we gonna collect this data? We all know we are on the DNA. Can you tell me 5 sources of DNA from where we can get the DNA? Hmm? I mean, the human samples. Human samples. Blood, cells, skin. Yeah, you all know better. So where all this information is collected and stored in the form of the databases which are actually present over the internet. So this is one few of examples, Unicron, IETD, PTD, HubChem, GEO. So these are the databases which from where we can take the data of our own interest. Thank you. Then we move on to features and generic. As being humans, we understand languages. You speak Telugu, I speak Hindi, we speak English. English is made up of 30 alphabets. So that's saying everything. Okay, I can't practical that in computer base. Okay, so let's say one. English is made up of 26 alphabets. The combination of these alphabets makes sense, right? So my name is Neelam. So Neelan. This stands for Neelam, correct? If I remove L and I put it in front of N, then the name changes. So the feature engineering is all about taking those specific information from either DNA or RNA protein and then feed this information to machine because machine do not understand English. Machine understands numbers. Machine understands numbers in the form of binary, either one or zero. We have to convert this information into numerics. How do we do that? The feature engineering has to do this. So there are two types of features, numerical features and categorical features. So can you move on, please? Yes, thank you. Next slide, please. So, yeah, as you see here, we can say that polypeptide of like set of many amino acids. So, how can we convert this information into numbers? Can you give me 5 different amino acids? RP, QC, you know that. So how do you convert these words into numbers? So what we do is there are 20 amino acids, we can find the composition, we can find the percentage, we can find like what amount of amino acid is present in a given protein. So this is how we compute the features. Amino acid composition, dipeptide composition, tripeptide, atoms and bonds, how many bonds are there? What kind of bond information are there? What is the arrangement of amino acids? So this is the information we actually translate from alphabets to numbers. This is how we generate the features and then yes, comes the data modeling. Data modeling is all about we should train machine learning models. Whatever the information we have given, first we have extracted the data, we have converted it into the numbers, and now we are training machine learning models. Yeah, next please. So this is one level which we use very frequently in bioinformatics that we do not give all the data to machines. What we do is like in our schools, what we have? We have upstairs, the teachers or the lecturers provide the lecture and then they take a surprise test. So we also do the same with machines. We do not feed the whole data. We provide them the 80% of the data and then provide the surprise test to the machine by seeing that is the machine has trained properly or not. Let's say I have trained my machine learning model on the data set of cats. So I have given a different kind of tasks to my model. And then to check whether the model has been trained right, I have now shown the picture of the dog. What do you think? The machine should categorize it as. If I train my model on the data set of cat, and I now give the data as dog, tell me what the machine should categorize it as. It should say, is it true or false? False, right? So this is how then we come to machine learning evaluation. Next please. That's how to split the data. These are the more critical points. We have less time, some critically points.
Next please. So we fill in the data, we evaluate it, correctly that it is false. So then we make our machine learning model day again. Say, hey, this is cat, this is dog, learn it. The next time I give you another model. This is how we say that we have to cover the data, train it again, test it again, train it again, test it again, until unless the model says accurately, say, hey, this is cat and this is dog. This is true and this is false. Next please. Yeah. There are different types of machine learning models available. Supervised, unsupervised, reinforcement model. Supervised models, unsupervised models, these are the names of the classification regression models. I'm not very much deep into them. It will be covered overall information for you. But you might have heard about support machines, random forest, logistic regression. And if you haven't heard about, we have not written anything. I'm glad to help you out. Okay. So yeah, then is our performance selection. How do you evaluate? As you said, that's the true dogs response. This is how we evaluate the performance. These performances are evaluated in the form of different parameters. Next week. This is called confusion matrix, which is in the form of true positive, true negative, false negative. So let's say now I have modeled the picture. Body of the dog with the face of a cat. Now machine learning model gets reduced to the dog or to the cat. Then we might predict it as false positive or false negative. So this is how we get the numbers and this is how we get the machine learning models parameter performance. Yeah, these are some of the critical terms which we use for performance evaluation. Next, please. Then the one, most important. So we have done everything else is well, it's ready, model is ready, data integration, we can integrate, everything is done. Now, what? We have to deploy the model in the form of anything, so that the people outside community can use it. So we can deploy the model in the form of mobile applications, web services, database. This is the basic machine learning pipeline. If you all have read it right, and if you all are not sleeping, so can you please tell me about that? Starting with data collection, then, and then a big calculator. So this is one of the real-time applications. It's what you have to produce the jelly. This is application in real time. So you all can say, whatever you tell me, Google can tell, Scientific can tell, shows the real-time application. So yeah, the real-time application is how I have used machine learning to identify allergenic proteins. And such a topic, starting to make up the place. We all know allergies. We all know allergies. Anyone who is non-allergic, anyone who gets the allergy, dust, drug molecules, we eat something, right? So that causes allergy. So it is very much important to address the allergy, right? Whenever we get fever, we take paracetamol. Have you ever come to report to the doctor and I'm not allergic, and then he gives you a treatment? So that's where this was one of the objectives I did during my PhD program to address allergies and physiology learning. The 5 basic steps. Trying to keep up the rates and see that how I have used all the right basic steps to address allergenic proteins. Next please. Number one was data collection. I collected the data. Collected the data from previous activities. I collected the data set from different databases, very broad analysis online and different literatures, especially. Then this is how all the data set. I called positive data set, these are the proteins, allergenic proteins and the proteins which are non-allergenic and negative data sets. This is how the data looks like. Proteins are made up of amino acids, long length of amino acids, more than 50 amino acid length is known as protein, so we have a various length of protein. Then second step was pure left. Different. So different has mentioned amino acid composition, dipeptide composition, base features, another touching point not doing much. Anyhow, what we can do is convert the alphabetic information to numbers. So there are various kinds of methods available online. How we can convert the alphabets to numbers. So this is here. Next please. You remember how the data reflects? The data was A, N, L, G, T, R, D, big length of protein.
And this is how the features look like. Can you see the numbers there? The 55, 8, 3, 30, what have I did? What I did was to find out that how much percentage allergy is actually present in the whole protein strand. That's how I converted that to number. Thank you. Then, third point was, data. So we have, there's a different data happening, data machine learning algorithms. Learn is actually online available, free available machine learning module. Anyone can use it, and I would highly suggest to go through this package to brush up your or to get the basics of machine learning algorithms. Right, please. Same fourth step was this is going basic. Performance evaluations. I have evaluated performance and saw that okay. Machine learning model called random forest is actually able to classify a protein as allergenic and non-allergenic. So when I train my model and when I get the unknown protein, my model was actually 92% or 98% correctly predicting the protein as allergenic and non-allergenic. So this is how the machine learning model works. And the last step was deployment. Thank you. I have deployed in the form of prediction server, web server, which is easily accessible to the environment. This prediction server named LG Predict 2, allergy protein prediction 2. It is the second method that we have made it as 2. And there are several modules. Here you can see that we can just place the protein and it will hit the run, it will give you the output. This is the output. It will tell you the protein and where it is allergen and non-allergen. So wherever experimental scientists like Dr. Ravi, he is working on when he was working on making the vaccine and the yogurt as a vaccine, he had to know whether the protein he is using as a vaccine is has some allergenic properties or not. If it does have allergenic properties, then it will be changed. What he can do is he can use my Excel. Okay. Use my Excel, don't forget to cite it, and yeah, the half job is done. So this is how you use the machine learning things in any data. For you people, it could be drugs, it could be DNA molecules, it could be chemical compounds. Because you people are much more do much more integrated science than I am able to do for computer science. Next please. So yeah. So this all has been finished. This machine learning pipeline I have integrated in the form of the server called edge question. Next. Why are we doing all these things? We as researchers have to publish our work. Is this work if we try to do these kind of problems? Can we get the publications? Yes. This is the work which has been published in mathematics. It is actually one of the prestigious advantage. And this work has been cited more than 200 times, which means people are actually using our work. So can you see the difference? That rather than testing each and every protein in animal models and in human models, we can actually use machine learning and mathematics skills to scale down all those testing only just a matter of few minutes. Yeah. So what has already been published? So now the question comes, why bioinformatics is a crucial skill for pharmacists? Now I would like to know your thoughts about it. Do you really think that bioinformatics is a crucial skill for you all? Or maybe you are yawning. Do you really think that bioinformatics is a crucial skill for you all? Still not. And next time I go better job to kind of say that. So to help the pharmacists to stay in a digital era, precision medicine, drug administration, and research and innovation. Next. These are the skills which you can learn. So when you gain these skills, when you know how to programming, Python programming, how to use the basic command line code, you can there bash the shell scripting, how to use the tools and databases and develop the servers and things. You are actually ready for the market. And when you go, be it your degree, B pharma, whatever. And you say that apart from my degree, I also have the mathematics skills, I can do this roughly. you will get a very good job with a good package. So if you are interested in good packages, so yeah, you have to learn mathematics skills. The next please. So these are some of the databases, we can discuss it in a one-on-one interaction and have one-on-one interaction with you all. I can explain that time. You take it back. Thank you. These are the career opportunities. We can easily play with pharmaceutical companies, the TX, and yeah, academia for startup, as Dr. Ravi has his own startup. So yes, these are some of the. Yeah. You need to train yourself rather than being strict to that we are pharmacy students, we don't know how to use computers, we can't do that as skills. You have to train yourself. Let's integrate that into digital pharmacy. And then next is how can you get started now? How can you get started? Start from basics. So then you can go to different courses, there are very online certification. One of the courses has been delivered on course at the University of Michigan. It's wonderful course for Python for various. I would suggest everyone of you go and have through that course. And so machine learning, and learning, networking and internship. So yes, I have the students who are from pharmacy, biomedical and computer science working along with me on the various different projects. So I'm also looking for good students. So if you think that I have convinced you that I'm part of the good student, you can approach me. Thanks. Yeah, the conclusion is yes, bioinformatics is actually shaping the future of the pharmacy and learn bioinformatics and stay ahead in your career. These are some of the topics which I discussed with you in one-on-one interaction, which you can like how can you start using these capabilities. Next. Next, please. So we can read my publications, which actually have developed learning methods and developed various databases and methods with my Google knowledge. And this is the lecture that I'm always keen to deliver lectures and to address good students and indeed future researchers like you. These are some of the achievements I have. So I would like to highlight this administration and the science leadership program. I was one of the 100 women leaders in working in science area, and the DST has provided us the training, 4-day training. I was driving a lot, so I take pride that I represented my college there. One of the 100 women scientists in India. So yeah, then we are then we wanted to thank the Overall campus for actually inviting me and sharing this page with all the wonderful audience. And then I'm just working in this area, whatever the work I have done during my course of work, PhD work, thank you all. We also want to we can email to me anytime if you have interested in doing some of the content, you can send me over email and you can read my research to Google Scholar. Thank you so much. So are you ready to begin your career in shifting yourself as digital pharmacist? I didn't hear that. Congratulations, dear. Thank you for being a wonderful audience. Open to all the questions.
Bioinformatics are computer systems to be the syllabus of pharmacy. Because now most of the colleges have become autonomous. They are having a choice of around 20% they can vote in. Unless this will be as if you are saying querying about the mathematics. So when it comes to terms, there are platforms are coming. You can suggest some of these courses. The main for the autonomous are just to introduce the computer courses. Not simply on learning computer technology, which is more relevant to the pharmacy, how you are developed to disease.
Okay, so you are asking me to test all the courses which are relevant to pharmacy, right? So, to be very honest, there are no courses which are relevant to pharmacy.
Not relevant for that. They have started working with the pharmacy and then developed in the bioinformatics.
Yeah.
It requires a lot of skill, but they have to enter into that pharmacy. You have to make them to enter into that course.
Yes, because now we need to learn the basics. That is nowhere related to pharmacy, nowhere related to biotechnology, nowhere related to bioinformatics. That's just the skills that you can gain by learning the basic programming, basic bash scripting, making friends with computers actually, this is very much important in current scenario.
Unless you make it compulsory to the students, intentionally don't come. We are having number of courses in the campus. Unless you push them to join, some people are hesitant, they will postpone. You should make it one of the subjects compulsory. I tell you, don't.
But I believe they are good machine learning models, whatever the data I have given them, they are trained well. So I believe that if these kind of courses are provided to you, please take this opportunity and learn and upskill. Trust me, guys, when you start out of the college and when you go to the recruiter, it is not going to see that whether you have gained whether first rank holder or what. If you bring what the recruiter needs, you will be hired. So please upskill, and as Sir said that there are courses which are included in the campus, please go and attend those courses, learn these and learn these skills and learn the go.
Thank you, ma'am. Thank you. Next. So, ma'am will be here, okay, those who are, we are running out of time, the last session will be started in a few minutes, okay? So, next, please.
Thank you. Thank you, ma'am, for providing us insight on machine learning and bioinformatics in pharmacy. Now, I call upon Dr. P.S. Nagaraj sir, and Senior ma'am, and L.J. ma'am to come on to the dais and honor Neil ma'am. Thank you.
See further, there are scientific presentations of honoring speakers. Dr. Vasundhara Banraji Ma'am, Assistant Professor, Department of Pharmaceutical Information, NICL, Hyderabad. Now I call upon Nandini to give a brief introduction about ma'am. Good afternoon, everyone. These story and model are related to review and distinguished academicians, researchers, and mentors who have been shaping the future of pharmaceutical sciences with our expertise and dedication. Dr. Vasundhara Banraji, Assistant Professor at National Institute of Pharmaceutical Education and Research, Hyderabad, serves as the faculty in charge in the Department of Pharmacoinformatics. Her academic journey speaks volumes about her excellence. She qualified GATE and GPAT in 2007 and 11, along with clearing the faculty eligibility test at JNTU. And speaking of research, she has made remarkable contributions in the field of infectious diseases and drug discovery. Her work has been widely recognized with multiple research papers and have been cited by scholars across the world. Some of the highly cited publications, computer-aided design vaccine, artificial intelligence, and machine learning in drug discovery. A comprehensive review which is published in the European Journal of Pharmaceutical Sciences with over 100 citations. And it is multi-person tolerance in clinical isolate of Leishman, which has over 95 citations. Such remarkable work does not go unnoticed. Dr. Vasundhara Banraji has been honored with Best Research Award by the Indian Pharmaceutical Association in 2022 and Young's Young Scientist Award by the Indian Pharmaceutical Society in 2020. Since She has also an esteemed member of various professional organizations like Indian Pharmaceutical Association, Indian Pharmaceutical Society, and Association of Pharmaceutical Teachers of India. With such stellar career and immense knowledge, she continues to inspire and lead in the field of pharmaceutical science. Without further delay, let's welcome the president of the art, the scholar, the researcher, and inventor, Dr. Atul Kandari. Let's give her a huge round of applause.
So good afternoon, everyone. Am I audible?
Yes, madam, you are audible.
Yeah, okay. So a very good afternoon to all the audience and the organizing committee of Promilan 2025, and a special mention to Professor Dr. K. Natraj sir, who accepted my request to go and give an online talk. I would have loved to come there, but due to some other meeting on the schedule, I couldn't. But hope to see you all in person by next time. So I will just share my slides. So is my screen visible to you all?
Yes, madam.
Okay. So as the topic of today, Promilan 2025 is a very interesting and a topic which very close to my heart. So it's about revolutionizing the healthcare and taking in factor about the pharmacy, bioinformatics, and the health information technology. So my talk in the next half an hour and so, we will be focusing more about the bioinformatics and its application, that how from 20 or 30 years back, how it has revolutionized the healthcare and the patient care management. So when we talk about any revolution in healthcare and bioinformatics deals with the major biological macromolecules. Those are DNA, mRNA, and proteins. And this is also referred to as a central dogma of life, where DNA codes all the information that what could be translated into mRNA, and mRNA ultimately leading for the protein synthesis. So when I talk about DNA is actually nothing, just a blueprint of our life. It encodes all the information that how our body is going to develop, what are going to be the prone areas in our body, which disease we can be prone to, and how our future is going to be. That genetic code has several hidden codes that needs to be decoded. So that's decoding of that DNA and our blueprints is a main factor in bioinformatics. So bioinformatics is one such stream which is cannot be done solely. See, bioinformaticians depends on other fields, other researchers, and other researchers also depends on bioinformatics. So it is like a give and take, a symbiotic relationship. The major use at present is the computer scientists in drug discovery, chemistry, pharmacogenomics, infectious disease, and structural biology. In all those fields, bioinformatics play a very crucial role. Now let's have just a brief history how this field have emerged. So it is not a very older field of science or research. It started in 1965. But at that point of time, for the researchers, that was a quite tedious job. Because that time we don't have a very good computing power. We don't have that kind of computer scientists which could help us to decode the things. So the first initiative was taken by a lady scientist, Margaret, in with Richard. So what they have done in that they have compiled all the data manually about the protein sequence and structure, which were present worldwide. They call upon everybody, manually compiled a book and publish it. So that was the story back then, when we don't have such computing power, no networking. Now we can share everything on WhatsApp, emails. Just in a milliseconds, we get all the information. But hats off to her that during that time, anybody who was interested in drug discovery, they needed a protein structure. So she compiled it. So here it begins the journey. Until 1999, everything was moving on the fast pace. So when we talk about the fast pacing, the very good example is the Human Genome Project. So earlier, the Human Genome Project initiated in 1990, and it took around more than a decade to decode our human genome. A single genome decoding took $1 billion of money and a long span of time. But now, with the high computing power, the more an understanding of the data or the genes, how it is working, we can do it in a span of a day. approximately costing around 10 to 20,000, I can tell you what your blueprint or what genes do you have, what mutations do you have. So this human genome project have really revolutionized the era of bioinformatics, which led to improved clinician decision, patient management, and drug discovery, and open several new avenues. That is the CRISPR and all has been, we are all grateful this is a human genome project. So now when we talk about the benefits of this human genome project, now when we decipher the blueprint of a human genome, now what other thing we can use it, how to use that information, that there comes the bioinformatics. So we have different subdomains in that. We work on genome. Genome is just an information of the DNA, just the sequences in which we only get the information about the mutations or the copy number variations. The other, here's the transcriptome. As I mentioned in my first slide about the central dogma of life, about the mRNA. That works only about the gene expression studies, that what genes are up and what genes are down, because DNA will only tell about the sequence. Then after going through transcriptomics, a new field of epigenetics, because we all know that the environment really affects how our body responds, whether it's a disease, whether it's to any allergies and all. So in those cases, these studies become very important because everything is not about genome or transcriptome. It is also about how our body is responding. And in case of epigenetics or epigenome, there is no changes in the DNA sequences. It's just about how it affects the gene expression. Then cellomics is there. We talk about the cells and what is happening inside a particular cell. Proteome and metabolome, as the name suggests, it's about the total protein profiling. And the new subdivision of that is metabolomics. So our body produces different sets of metabolites. When we take a drug, different metabolites are produced. When we are diseased, different metabolites are produced. So these metabolites also set us apart. In some cases, detection of a certain metabolites in our body can predict what kind of diseases is there or whether we are able to metabolize a particular drug or not. And other important factor which has come in place, microbiota, which has been very popularly studied, and too much research now is in a neurodegenerative diseases, where we have heard about this common term about the gut-brain axis. So these microbiome, these microorganism which survives in our gut controls how our gut behaves. It produces metabolites, which affects the epigenetics, which affects the DNA. It may cause mutation. So these were the major subdomains of bioinformatics data analysis, which currently we use at NIPER and other researchers all across the world to understand or to identify the new drug targets, to identify the new vaccine targets, or to look for and design the new drugs. Thus, further refinement is single cell sequencing, microRNA, and genomics, jivas, all those studies are there, which tell us further refined information about a particular parameter if you are interested. Now, how it has helped us. So if, for example, I take a person who is healthy and a person who is diseased, so what is going to happen? By just seeing it, the doctor is just saying that, okay, you go for this blood test and come back. But if we are going to do some new disease has came and we want to identify that what could be the new vaccine, how we can design a new vaccine, what could be a new drug targets, disease biomarker. So all this analysis of the genome, whether it's through genomics, epigenomics, transcriptomics, will help us to identify, and it will certainly fuel the discovery of drug discovery as well as vaccine and drug targets. So this is an overall representation that what happens, how the data is being analyzed, and at different points, we can see that we can combine all these technologies and try to get down and narrow down to a particular subset or a particular research question which we are interested in. So in all these aspects, data analysis and bioinformatics plays a very important role, because just having the data out of the machine, nobody can analyze it. Here comes the power of bioinformatician who tell us with the help of a core biologist that what this gene does and what it possibly can do. Now, one more example. So I have just taken few good examples where I can correlate how bioinformatics is helping. So one of the very crucial example, which is very close to everybody nowadays, is antimicrobial resistance. And I'm sure all the students, the B Farm and M Farm might be knowing what AMR is. So we all know that if a doctor prescribe us a current antibiotic and we are not responding to that, means that bacteria you're not able to kill. It is not bactericidal, not bacteriostatic. Then what happens? After 5 days, you complete the course, you again go back and say, my symptoms are not subsiding. Then again, he will prescribe you a new drug. So with this series, when you are still non-responding, then you will come to know that, okay, the bacteria with whom I was infected or with whom I am still infected is antimicrobial or a multi-drug resistance antibiotics. For example, TB. We have heard about MDR, XDR, and total DR, mean extremely drug resistant, totally drug resistant, and multi-drug resistance. So the current scenario in the hospitals, which they use is they culture the bacteria, then they go for the phenotypic characterization. That is by the simple disk diffusion assay or a microblot dilution assays. But in that case, the time they take is around 96 hours for some slow growing bacteria. And till the end, we come to know sometimes the disease has progressed so much that it's very difficult to save the patient, especially in case of the bloodborne sepsis cases, where the patients have only 4 to 8 hours to tell us what drug. So in that case, we just have to dump all the antibiotics into that patient in order to save us. So now how this new computation analysis, how this new next generation sequencing system can help us. So if I just take the blood of that patient, isolate the DNA and sequence that, I can detect what kind of bacteria is it. I can detect and check whether what kind of antimicrobial resistant genes are present in that. So what it will do, it will just drastically reduce our detection time. It will get that hope to that patient family as well as to confidence to the doctors to make the treatment accurate so that the patient is cured without much of the side effects. So this is just an example how the workflow look like. The samples are there. So we can directly go for the biological samples analysis, or we can isolate the DNA after culturing and then analyze. So this is done usually when we have a reservoir of bacterial samples in the hospitals to do a surveillance studies and that what is going to happen and what kind of pathogens are being present in the hospital. So after the fragmentations, the machine give you an output. This is ATGC sequence format only. Then it is the role of the bioinformaticians or the data analysis person to narrow down what this information codes for. COVID-19, we all have suffered a lot. So why do you think, on what basis the variants were named alpha, beta, gamma, delta, and omicron? The genome of this SARS-CoV-2 is like this only. You will get few proteins, nothing much, not complex, but still it had make our life hell. It has led to the more number of fatalities, morbidities. The only reason why these variants were named was depending on the mutation or the sequence changes that were observed in this part, the brown highlighted part, that is the spike protein. On the basis of the mutation that we have found in them, they were labeled as, and the basis of the mutation they did, the symptoms also vary. We all know that a delta has resulted in a lot of loss of lives. However, Omicron was very fast spreading, but it was just like a normal influenza infection. The doctors has advised to stay at home and let our body give good rest and fluids. And with the medication, we were able to cure. The other important parameters which help us to understand COVID-19 and to tackle is using a different omics profiling to understand what will happen when a patient is infected, why one patient is asymptomatic. In the same family, I have asymptomatic, I have mild to moderate, as well as I have a severe case of person. So all these answers were given by analyzing the data, what we obtain from different omics analysis. Another example of how this has revolutionized the patient care and the healthcare is cancer. So in this figure, if you say it is talking about the esophageal squamous cell carcinoma. So what therapy? Because we have heard a lot about that cancer treatment is not working. there are resistant cancer cells infections, there are relapse of infection. That means that a drug which were given to tackle that particular cancer cells was not effective. So in those cases, they have divided them, analyzed their whole genome, and with different techniques like whole genome sequencing, whole genome bisulfite sequencing, RNA-seq, single cell RNA sequencing, and proteomics, they found that in all kind of these patients, cohort of patients, there were changes in the immune modulation, the cell cycle pathway activation, NRF2 concogenic activation, and immune suppression. So in the patient in which immune modulation is high, they will be responding to the ICB therapy response. In the case where the cell cycle pathway was activated, we have to give CDK4/6. So how this information will be given to the clinician? Only after analyzing the data. Otherwise, they will be just put on a single kind of therapy. And after losing so much time of 2 months or 3 months, then we'll come to know that, okay, the patient is not responding. So in those cases, this kind of analysis of the data, the bioinformaticians plays a very crucial role. This is also a similar kind of example. So now, after understanding how the data is, there is a very new era of bioinformatics. A new field is referred to as immunoinformatics. Now, not too new, as during COVID, it has been expanded a lot. A lot of research, a lot of money has been kept. So, as the name suggests, immunoinformatics, immunology plus informatics. You know about immunology, students? It's about the B cells, T cell, our human immune system, how we defend our system against several pathogens. So what if without culturing, without doing any wet lab, we have a prediction to make? So this field help us to understand the approaches and predict the immune system responses, contributing to designs of novel vaccines, immunotherapies, and it can help us in study of several immune-related diseases. I can have the DNA sequence of a particular patient. I can have the sequence of the virus particle. I can dock them with each other. Means I can check whether there are some complementation of them with each other. If it is, then I can expand it. For vaccine, what we know, we want a region of that pathogen, which on acting on that human surface or on the human body, will elicit some immune responses. So broadly, immunoinformatics have been playing a very crucial role in disease prevention, disease treatment, prognosis and monitoring, drug discovery, of course, and disease pathogenesis. So when talk about disease prevention as a whole, because it is more well understood, we talk about vaccinology, structural vaccinomics, allergy prediction, and host pathogen interactions. When we talk about host pathogen interaction, why you have seen in COVID or some diseases, when in layman term we talk about a weak immunity and a strong immunity. It is nothing but how the host and pathogen are interacting with each other. Are the body is strong enough to tackle that infection, or is the pathogen strong enough to model our system against us? So that is host pathogen interaction, and all this information is hidden in the genome and the interaction pattern between the host and the pathogen. So when I talk about this reverse vaccinology, what is been used in this is, I have a cohort of pathogen or any single pathogen. I will sequence their whole genome, and I will find those areas which are similar in all those genome. Okay, so what happens? These genome which are sequence which are similar, I can take out that and I can check whether they can elicit any kind of human immune responses. This is all done in silico, means in at the computer level. Then when I get a good lead or a good hit which predicts, yes, when this sequence of the pathogen is being inserted in the host, some immune elicitation is there, then we can synthesize and check. So in the long run, it is reducing the time. In COVID, we have seen that vaccine was developed in a very quick time. Why? Because we have very advanced tools, we have very advanced bioinformatic analysis, which help us to make the vaccine in a short span of time and save the human race. So this is the example of what happens and why they have predicted that ACE receptor, we have seen, they identified that the virus particles bind at the ACE receptor. That's the spike proteins. So they have sequenced it and identified which can bind to what. So that's why everything which can block this can help us in inhibiting that infection. And in this, this is a different sequence analysis of various COVID strains, alpha, beta, gamma, delta, and omicron. All these red particles indicates the mutation in the spike proteins of that. So that lead us to very crucial information that where the mutation is happening and what could happen if this mutation is occurring. So all this thing at the level of the computer, you can predict and see what can happen. So for this, I would just like to add a few things that sequence analysis is very important. It helps us to understand, see, in case of COVID, what happened? We have no information how COVID is going to react, how COVID going to impact our body system. But we know that with the help of the phylogenetic analysis or the genome sequencing, that it was somewhat similar to the other pathogens previously which has infected. So with the help of this, we were able to match the sequence between the COVID and other viruses and try to understand that what could be the outcome of the disease, which also helped us to analyze and predict what treatment could have been given. Now, another important thing which we all have seen in your books also, the drug discovery process. So why do you think traditional drug discovery, or do you think the traditional drug discovery process is good? Does it has any drawbacks? Yes, of course, it has certain drawbacks. If you talk about this, the research and the development span 6 to 10 years, then preclinical studies, of course, 1 to 2 clinical trials, review and approval. So this method is quite tedious, expensive, and time consuming. Now, what can be done to reduce this all? So that is now referred to as a modern drug discovery process. So all these steps before the preclinical trials, which were earlier showing a span of 6 to 10 years, can be reduced to 1 to 2 years and then can lead us to a very quick identification of a new treatment option. So all these parameters, you can just sit in your lab and analyze it if you have a good understanding how to analyze the data, and it will give you a good output. So when we talk about, just for students' sake, I'm just putting, when we talk about this docking study and drug discovery, it's a simple lock and key effect. So whatever protein, the active site is there, if our drug can bind to that active site and can give us a therapeutic effect, that drug target interaction is good for any pathogen. So this is just a brief overview about how we work. There are two parameters. Either I have to know the structure of the protein against which I am designing, or other than that, I know the one chemical compound which is very active, and I want to identify something which is similar to that. So that are two strategies which is commonly being used in computer-aided drug design. That is, the one is structure-based and ligand-based. So this is one database which is freely available. That is a protein data bank. If you want to study any of the drug molecule and you know the target in mind, you just have to put your requirement here, whatever the protein name, and put a search command. You will get to know that there is that protein structure is known or not, because we need a starting protein structure for that. So this is a very freely available tool which anybody can use. I think students might be aware of that. But what if you don't have? What if the structure is not available? So these are other methods like homology modeling, threading, and ab initio method, which can be used to determine the structure at the computational level. So homology modeling has a requirement of some sequence similarity between any of the protein of interest and the target. Threading, if you don't have any information, then thread that thread piece by piece. There are tools which can help us. Similarly for ab initio, but for the savior of the students, we have AlphaFold now. It is an AI-based protein detection, protein structure determination tool, and it is 99.9% accurate. If you go to UniProt site, you will able to see this AlphaFold, and it is very useful. We have also used, and it is going accurate result as compared to the protein structures which are available with us. So this is what I have discussed about docking. I will not go into detail of docking here. Okay. So what are the major advantages of this modern drug discovery and how it has helped us? So these are the few of the drugs which has been very easily and has been reached past the clinical trials, has been used for treatment, which are have used all these techniques in combination to reduce their time. There is one more drug which has came for has been FDA approved, that erdafitinib, and it has been work of the collaboration between academia, biotech, and pharma. So all of them have used these techniques to shorten the time span required for a drug discovery. So this is one of our paper, just for example, I am citing. So as we know, COVID was there, but we were not future ready. Similarly, when we have seen few reports of monkeypox virus here, what we thought, what if it also become a pandemic? So we analyzed the genome of this monkeypox virus, and we identified with the help of the genome sequence and the data reported, we found that one of the protein in this monkeypox virus, E8 protein, is important because it is a surface membrane protein involved in the viral entry and adhesion. So what if I can design or identify few drugs which binds to it? So we have chosen few of the libraries which are the natural product libraries obtained from different bacteria and fungi and approved FDA drugs, and we have identified few hits. However, we are not able to validate it in vitro because we don't have the virus with us. But anybody who has this facility is free to use these data. It is freely available on website and can have the benefit of it. The other aspect of modern drug discovery, repurpose. So what if I have one drug for cancer and I want to try it for other infection? Yes, of course, it is possible now. That is referred to a repurposing. In COVID, it has been used. One of the articles have cited that they have started with the Campbell database, narrowed it down 38, and they have got very minor 4 to 5 major hits, which they are working on. So with the help of this modern drug discovery, now we can explore different strategies and we can get the desired results in a short span of time. Similarly, when we talk about antimicrobial resistance and we say, oh, this mutation has happened or the pathogen is resistant due to the mutations, what if I can make those mutations in silico in my computer and predict whether the drug is going to bind or not? So all this study I'm just referring so that you can know what it can be used to make ourselves a future ready. How data analysis and bioinformatics help us to tackle the problem which we might face in the future. This is also one of the studies by us where the protein structure of this FTSZ protein in bacteria was absent. We have predicted its structure using homology modeling and have got few good inhibitors who are giving us good hits, and we want to take that paper forward. Now, very interesting and the current topic is pharmacogenomics. Pharmacogenomics, as the name said, pharmacology plus genomics. It talks about why one patient treatment is not good for all, right? So for especially in case of diabetes, the why metformin, some people are responding, some are not. Just for example, I'm stating. So on in all these cases, the information is hidden and we want to identify it. That could be the CYPs genes, which are known for metabolism. That could be the efflux transporter genes, which efflux the drug out. But these studies have taken a lot of pace and people are working on it. So if there is a patient group present, we did the test, we got it. Same standard prescription dose for everybody. But if I combine the genetic test, I can understand the person blueprint. I can decode it. I can tell what kind of dosage can be suitable for them. What it does, how it revolutionizes the current healthcare system, it helps the clinicians to provide the best treatment. It helps the patient to be safeguarded from the several side effects. So in this way, it helps a lot. So this is all about how the healthcare system has been revolutionized with the help of bioinformatics. This is my core team, which is a combination of my MS student as well as my PhD students. And I think I could have done, I am able to do justice to this topic. I thought with because of the students are there, let me give my presentation with more of the examples, which with that they can relate. Thank you very much. If anybody have any question, I am happy to answer.
So any questions from the audience, you can just. Any questions from the audience?
Or you are shy to ask now, you may please mail me, and any students have any queries, I am happy to help.
Sure, madam, sure.
Yeah.
Thanks for the wonderful talk.
Thank you, sir.
My follow-up on Dr. S. Anandikumar's relatively vote of thanks to the monster.
It's my privilege to propose a vote of thanks to the morning scientific session today. So firstly, I thank our principal for his constant support and guidance. We thank Dr. Promilan, Associate Professor of the University, Department of Hospital Chemistry, for his insights in the post-pathogen genome comparison, identification of possible drug targets. So we are very fortunate today. So all the topics are very relevant to today's scientific community. So thank you, sir. We are grateful to Dr. A. Ravikumar, Professor in Practice, Andhra University, for his talk on evaluation of on-product induced mutations in human cytochrome P84 proteins. We would like to acknowledge our gratitude to Dr. Neeraj Sharma, DVIT Hyderabad, for a valuable talk on digital pharmacist in biotechnology, machine learning, and bioinformatics in pharmacy. We also extend our sincere thanks to Dr. Vasundhara Bandari, IPA Hyderabad, for a talk on antimicrobials and role of informatics in healthcare. So we thank all the speakers today in the morning session for their valuable time and also their wonderful sessions today. I think all of us have a great learning experience in the morning. So scientific sessions will continue. So presentations will continue now. Thank you.
Thank you so much for the participation.
So thank you, madam. Thanks for your presence.
Yeah, thank you. Okay.
Thank you. Thank you, madam. This is Dr. Nathraj.
Thank you. Thank you, sir.
Thank you.